Swayam Prabha. Digital India. Educated India.
Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we started talking about data normalization methods. We talked about some of the methods that address this gene length bias as well as the sequencing depth variation, and then we talked about between sample normalization. So, we will continue on that in this class and we will discuss more sophisticated normalization methods. Okay, so, this is the agenda for this class. So, we will talk about between sample normalization and then we will talk about batch correction. So, in between sample normalization we have already discussed two methods. So, the first one is median normalization, the second one is UQ normalization, and we have also seen their drawbacks, right. So, one of the drawbacks is that they assume that their count data distribution they differ by a constant across samples, and this does not hold in most cases, and also they do not address this RNA composition bias. Okay. So, in this class we will talk about different types of methods that can perhaps address these issues. Okay. The first method that we will talk about is called quantile normalization. Then we will discuss about median of ratios method. It is also sometimes referred to as or called as relative log expression or RLE method. They will give you identical results, and then we will talk about TMM normalization, and finally, we will talk about normalization using control genes or spiking controls. Okay, so, let us start with quantile normalization. So, what is quantile normalization? So, again it starts with an assumption. Okay. So, the assumption is that the read count distributions are identical across samples. Okay. So, this is a specific assumption, right. Maybe their genes are not showing same expression, but the distributions are identical. Okay. They will not change between samples. Okay. Or what it means in other words is that distribution of gene expression levels are identical. Now, if you think about this assumption, you might have some questions, but keep those with yourself for the moment, right, because we will discuss a bit more about this. So, when you assume that the aim of normalization becomes that we want to make these read count distributions identical, right. So, if we assume okay, they should be identical, but in reality they are not. So, we would have to do some sort of transformation so that these distributions become identical, right. So, that becomes the aim of this normalization method, and this is a normalization method that was employed for microarray, and it has been also adapted for RNA-seq data. Okay, so, what are the steps for quantile normalization? So, the first step is rank genes by expression levels in each sample, okay, and then calculate average values of genes of same rank across samples, and then replace the original values by these average values. Okay. So, we can actually take an example and we can see how these steps are executed and the results that you get at the end. Okay. We go back to the same example, right, because we are already familiar with this. We have the genes ABC, we have the samples sample 1, 2, 3, we have the raw counts. I have not given the total because the total is not really important here. Okay. So, what is the first step? So, we have to rank genes by expression, right. You can do low to high ranking or high to low ranking, right. So, here I have done high to low ranking, right. So, the letters and the numbers that you see. So, R1 stands for rank 1, right, R2 stands for rank 2 and R3 stands for rank 3. Okay. So, I have ranked these genes, we have only 3 genes, so it is easy, right. So, R1 is the rank 1 gene with highest expression, okay, and again we have sample 2, again rank 1 with the highest read count, okay, and sample 3 we have also done the ranking. Okay. Now, the next step is calculate the average values by rank. Okay. So, for rank 1, what is the average value? Okay, so for rank 1 we have these 3 rank 1, right. So, this is the rank 1, this is rank 1, this is rank 1. So, the total count is 6 plus 12 plus 7, right, and the average will be that divided by 3, right. So, for rank 1 the average value is 8.33, and similarly we can calculate this R2 average value and R3 average value, right. It is following the same process. We are calculating the average expression values by rank. Okay. So, this is what we get at the end. So, what is the next step? So, we have to replace the original values by the average values. Okay. So, average values of that rank. Okay. So, again if we go back, right. So, this was rank 1 and we are replacing this the actual value that we the earlier value, right, with this 8.33. This is the average value for rank 1. Okay. And similarly, here we have also replaced this with 8.33 because this is rank 1. Similarly, here we have replaced this with rank 1 again 8.33, right. Now, what you see at the end of this normalization, the distributions are identical, right. You see this, right. For sample 1 you have 3 values 8.33, 4.332. For sample 2 you have 8.33, 4.332. For sample 3 you have 2, 8.33, 4.33, right. The values are identical, but the genes may show difference in expression, right. That is what you see in this very small example. Okay. So, we follow this process for thousands of genes, right, and we generate identical distributions of read counts for each sample. Okay. And that is what I am going to show you now, right. Here I am showing you the raw count data. This is simple box plot, this is not an RLE plot, right, that I have shown you earlier. This is simple box plot where we are looking at the raw count data distribution for again those samples, right. And you can see that these distributions are slightly different. You can see this. So, each dot here is a data point, right. These are the outliers, and you can see this, like, distributions are slightly different. Now, after the normalization, right, after quantile normalization, you will see the distributions will become identical, right. Even if you look at the points, they are at exactly same place. You see the median value is exactly identical, you have these boxes, these are exactly identical. Okay. So, they become identical because that is the idea of quantile normalization, right. We want to generate identical distributions for all samples, right. So, here you have 16 samples and for each of them you are getting these identical distributions. Okay. The genes may show difference in expression, but the distributions are identical. Okay. So, now this is a probably a question in your mind, right. Can you assume this, right, can you assume that the distributions are identical, right. So, again the answer is no, right. This may not be always the case, right. The distributions may not be identical because some genes may show very high expression and that value may not be present in other samples, right. So, you can imagine different scenarios where this assumption will be violated. Okay. So, we come to the drawbacks, right. So, the assumption that the distributions are identical often does not hold, right, because of different scenarios, right. If you have like just 2 genes showing very high expression, you will not have those values in other samples. Okay. So, you are trying to artificially trying to contain all those values by making these distributions identical. Okay. So, this is one of the first major assumption, right, and this is a drawback, right. And again we have not addressed for this RNA composition bias, right. So, if you go back to our calculation, you can see, right, we have not addressed this RNA composition bias, right. And if you compare again, it might appear, right, okay, gene B, gene C, they are highly expressed in sample 3 compared to sample 1. Okay. So, this again makes it unsuitable for this comparative analysis between samples. Okay. So, we come to the next method, which is the median of ratios method. Okay. So, this is a method that again does not assume anything, right. What it does is like a series of steps to actually address this RNA composition bias issue, right, and along with other issues, other biases that are present in the data. Okay. And sometimes it is also referred as relative log expression normalization, right, although this was not introduced by the original authors. Okay. But if you see this term RLE normalization, you can probably then fall back, right. This is the same method as median of ratios method. Okay. Okay. So, what are the steps here for this method? So, for each gene we have to first calculate geometric mean of count data across samples, right. So, this is a between sample normalization. So, we are looking at count data for each gene across samples and then we calculate the geometric mean. Okay. And of course, in some cases if you have 0 values, the geometric mean would be 0. So, for genes where we get the geometric mean to be 0, we just discard them for the subsequent analysis. Okay. So, this is the first step. The next step is to divide read count of each gene by the geometric mean. Okay. We will again take the example and we will see the steps, then you will understand this better. Okay. So, we divide read count of each gene by the geometric mean, and we calculate the median of the ratios that we get after the divisions in step B, right, and we use this median as the normalization factor for that sample. Okay. So, maybe this sounds very complex, but once we do the steps it will be clear. Okay. And so, we have calculated the normalization factor, and then we divide the read count, okay, the raw read count that we started with of all genes in each sample by the normalization factor of that sample. Okay. So, these are the 4 steps. We have the first step where we calculate the geometric mean for each gene. We divide read count by the geometric mean, right, and once we get some ratios, right, we get some values. We calculate the median of those ratios for each sample, and those median values for each sample will be the normalization factors, okay, for those samples. And using that normalization factor, you divide read count of all genes in each sample. Okay. So, again each sample will have its own normalization factor, and we will use that to divide the read count. Okay. So, let us now look at this, right, in action, and that will be clear. Okay. We come back to the same example, right. So, we have sample 1, sample 2, sample 3, again we have gene A, B, C, and the first step is calculate geometric mean of expression of each gene across samples. Okay. So, we come here to this table, right. So, for gene A we have to take these 3 values, right, 6, 12 and 0, and we have to calculate the geometric mean, which I have written on an extra column here. Okay. So, for this sample, because this sample 3 read count is 0, so this geometric mean will be 0, right. So, that is something you probably are familiar with, how to calculate geometric mean. For gene B, right, we have sample 1, sample 2, sample 3, we calculate the geometric mean, right. Again you know how to calculate geometric mean, you should be able to do this. For gene C, again we take these 3 values, we calculate the geometric mean, right, and you continue this, you do this for thousands of genes. If you have 10,000 genes, you do the same process for each of them. Okay. So, once we have done this, the next step is divide read count of each gene by the geometric mean now. Okay. So, we have to divide these counts by the last column. Okay. So, we have discarded the one with 0 value, right. Of course, you cannot divide by 0, and that is the first step, remember, right. We have to discard genes for which the geometric mean is 0. Okay. So, we are left with these 2 genes now, B and C, and for each of them, right, for each read count we have to divide by the geometric mean of that gene. Okay. So, for gene B the geometric mean is 4.93. So, we are dividing the raw read count 3 divided by 4.93, and similarly we are doing the same for sample 2 and sample 3. For gene C, that geometric mean is 3.14. So, we divide the read counts by this number 3.14, right. So, you can see this for sample 1, sample 2, sample 3, we do the same. Okay. Now, once we have done that, right, we got the values now. Okay. After the division, these are the values. What you have to do now is you have to calculate median of ratios for each sample, okay, which will give us the normalization factor, or sometimes also referred as scaling factor. Okay. So, you take median for sample 1, right, these ratios. For sample 2 we take the median, for sample 3 we take the median, and we get these 3 different medians. Of course, I will not take median of 2 values, it does not make any sense, right, but you get the idea. When you have thousands of genes, you take the median and will get some values, right. So, I have simply giving you an example, we get, let us say N1, N2 and N3 as medians for these samples. Okay. Once we get these medians, the next step is to divide these values, these raw counts, right, that we have started with by these median values, okay, N1, N2, N3. Okay. So, we can now forget about this table, this transform table, we go back to the raw count data, and then we can divide by this N1, N2 and N3. Okay. So, this is the next step. So, for each sample divide read counts by the normalization factor. Okay. And for sample 1 the normalization factor is N1. So, we are dividing these counts by N1. For sample 2 the normalization factor is N2. right. So, we are dividing by this n2 number here for sample 3, the normalization factor is n3. So, we are dividing by n3. Okay. So, once we have done this, we get the normalized count data. Okay. So, one of the things you probably realize if you go back to this table, right. So, median, if you imagine that for the sample 2 and sample 3, the median would be higher, right, compared to sample 1. It will be probably twofold almost, right, 2 times higher, okay, approximately, of course. Again, it does not make sense, but again if you kind of think for a moment, this will be approximately 2 times, right. So, n2 and n3 will be larger than n1, right, in this example, as you can see. So, this is approximately 2 times. So, what it means, right, if you now look at this gene B and gene C here, right. So, if n2 and n3 they are 2 times larger than n1. So, once you have completed these divisions, right. So, 3 by n1 will be almost similar to in value to 7 by n3, okay, and similarly 2 by n1 will be very similar to this 4 by n3 value. What it means is that we have now addressed this RNA composition bias, okay, that we were talking about for all the methods. Okay. By discarding this gene, right, where we have 0 value, right, we have calculated these geometric means, we have calculated these ratios, etc., and taken the median, we have now addressed this RNA composition bias here. Okay. So, this is something that is an advantage of this method. Okay. Okay, so, now we can answer this question correctly, right. So, do genes B and C show lower expression in sample 3 compared to sample 1? That is actually no, right. There is no difference almost in expression between these two samples, sample 1 and sample 3, right. So, you can probably see now, because the normalization factor n3 is probably approximately 2 times than the normalization factor n1. Of course, again if you have to do this for large number of genes, you cannot calculate just median, but just as an example this holds. Okay, so, again we go back to those early plots and I can show you how the data would look like after the normalization, right. So, this is before normalization, this is the raw data, right, again transformed because of this RLE method, okay, and then this is after normalization using this median of ratios method, okay. And here you see, right, again the medians are more or less identical and the distributions have changed, right. So, you can go back, right, these are the raw data distributions, and after this normalization you get this kind of distribution for each sample. Okay. Now, as you can probably guess, the RLE plot actually is related to this RLE term, right, relative log expression. So, when you do this make this plot, you kind of follow the same procedure, you calculate the geometric mean and then divide by the geometric mean, you get this normalization factors and then you get these plots. Okay. So, that is why they are kind of centered around 0. Okay. Okay, so, what are the drawbacks of this median of ratios method? We have seen that this can address the RNA composition bias issue, right. So, one of the major drawback of course is that there is no consideration for gene length, right. So, this means we cannot do a comparison within a sample, right. So, we cannot compare expression level of genes within a sample. Okay. So, this will require some other method, right, within sample normalization method. So, median of ratios method is strictly between sample normalization, right. So, we can compare across samples, but not within samples. Okay. So, the next normalization method that is also very popular is called TMM normalization. Okay. So, again let us look at this. So, what it does, it is the full form is of course trim mean of m values, right. So, this TMM, right. So, we will talk about what are these m values that you get. Okay. So, this method also calculates a scaling or normalization factor like the earlier method that we discussed. And so, what happens is that we now understand, right, that reads that we get for a gene or transcript, right, this count is not just proportional, is not just dependent on the expression of that gene only, right. So, it also depends on expression levels of all genes transcripts in the sample, right. We have seen this in our small example. Okay. And one of the assumptions this method makes is that there is no difference in expression of most of the genes between samples, right. So, if you take let us say 1000 genes, probably 80% or 70% do not show any difference in expression. Okay. So, this is an assumption in this method, and maybe 20-30% or so they show difference in expression between samples. Okay. Based on the assumption, this method calculates a scaling or normalization factor. Okay. So, let us see the steps, right, how it works. So, the first step in this method is you have to choose a reference sample. Okay. So, when you are comparing two samples, you choose a reference sample against which you will compare another sample, right. So, once you have chosen this reference sample, you are comparing against other samples and you can calculate something called M and A values. Okay. So, these M and A values will come later on as well for different purposes when you talk about differential expression analysis, etc. Okay. So, how do you calculate this M and A values? So, for gene G, right. So, this mg, right. So, these M and A values are calculated for every gene, right, that are present in the data. So, mg is simply log of this EGI divided by Ni and divided by the EGI prime divided by Ni prime. Now, this EGI, this is the read count or expression of gene G in sample I, okay, and Ni is the total read count of sample I. Okay. And the lower part, this EGI prime and Ni prime, this is for the reference sample, right. So, in the first step you choose the reference sample and then you get all these values. Okay. Similarly, you can calculate this Ag, this is kind of an average expression, right. So, again you have this half log 2 EGI divided by Ni times EGI prime times Ni prime. Again we know that EGI is the read count for gene G in sample I, Ni is the total read count, and then the similar measures, right, we have EGI prime and Ni prime, this is for the reference sample. Okay. So, once you have calculated this, right, so the next step is to trim these M and A values and it's usually extreme 10-20% or 30% values, and so trim out those genes that show these extreme 10-20% values, right. So, you get a distribution and trim out these 10-20 or 30% values. Okay. And what it means, you are removing genes that are showing differential expression. Okay. They are showing difference in expression between samples and you are removing those genes before you calculate the normalization factor. Okay. Once we have removed these genes, then we calculate this weighted mean of trimmed M values. Okay. So, we do not take these extreme genes, right, and their M values, we just take the filtered M values or trimmed M values as we call them and take the weighted mean. Now, the question is what is this weight, right, how do you calculate this weighted mean. Okay. So, weights are usually inversely proportional to the variance in read count, okay, and this is to avoid, right, genes that show a lot of fluctuation between samples till even after filtering, right, they should not bias the mean too much. Okay. So, that is why the weights are inversely proportional to the variance in read count. And again the idea behind this is the genes that show constant expression across samples, right, they should have the highest weight. Okay. They are clearly not differentially expressed. So, they should have the highest weight in determining the normalization factor. Okay. And this weighted mean is then used as the scaling or normalization factor. Okay. And so, you are comparing between two samples, what you will end up with is a one scaling factor for comparison between these two samples, and you can then use this scaling factor to transform this data, right, reference as well as the non-reference sample. Okay. If you are comparing between multiple samples, we choose one reference and then we can calculate scaling factors for each pairwise comparison, right. So, in that way you can handle these multiple comparisons as well. All right. So, let us again go back to the example, right, this is the real data set. So, again before normalization, this is the distribution, these are the distributions for these samples, 16 samples that we have, and this is after TMM normalization. You can see this, right, the distribution, the medians become identical, right, and again the values kind of come close to each other, right. Okay. So, we have seen this, right. So, just to again give you some something to think about, right, here is the raw count data in our case, right. So, you can think about how do you proceed with about this TMM normalization with this data set, right. We will not do this, of course, it does not really make sense to do this on a very small data set, because again you cannot throw out extreme values, but you can at least think about the steps yourself, right. So, for example, we can choose a reference sample, we can calculate those M and A values, right, and then after discarding those extreme genes, right, we can then calculate this weighted mean, etc. Okay, so you can think about the steps with a very simple example, and you will get this idea, right, behind all those complex formulas, etc., that we used. Okay, so we will talk about the next normalization, and this is quite different from what we have discussed so far. Okay. So, this normalization method uses something called spike-in controls. Okay. So, what are these spike-in controls? So, spike-in is an external RNA molecule that is added to RNA-seq samples. Okay. So, these are not part of your sample, right, you do not isolate them from the tissue or cells, right. These are synthetic molecules and external molecules that we add to our samples. Okay. The question is, why do we add them, right. So, again there is a consortium that actually has designed these spike-in controls. These are synthetic RNA molecules, right, and kind of taking care that these are synthetic, they do not show any similarity with RNA molecules that are present in organisms, etc. Right. So, you will see this term ERCC spike-in controls or ERCC spike-in mix quite often, and you now understand what this mean actually. Okay. So, this is usually a mix of synthetic RNA molecules of different sizes, and these are also mixed in different numbers. Okay. This is not a single molecule, you have many molecules like that, and they are of different sizes, lengths, right, and they are mixed in different numbers, right. So, just to see like how they are actually come out after you do the RNA sequencing experiment. Okay. So, how does it work actually? How does this method work? Okay. So, what we do in this case is that we add equal amount of spike-in controls to each sample, right. So, if you have 16 samples, we add this equal amount, exactly equal amount of the spike-ins to these samples, right. So, there is no biological variation here, okay, because we are adding this manually, right, after the RNA extraction. After we have purified the RNA, we add this and then we do the same steps, right, for the library preparation, sequencing, etc. Okay. So, any variation that we see in the read counts, right, that will arise purely due to technical variation. There is no biological variation here. Okay. It will purely come from experimental steps that we are following. Okay. And using this control, we can now normalize for all sources of variation, technical variation, using generalized linear model, right. So, we can set a generalized linear model, and researchers have done that. There are packages, we will see these things later on when you actually do the hands on, we will actually do this process, right, and we can remove these unwanted technical variation. Okay. So, I will just show this data again, right, because this data set had these ERCC spike-in controls, right. So, you can see this here. Okay. Again, this is the raw data. You will again see the RLE plot, right, again for all the samples, and this data had ERCC spike-ins. You will be able to see that if you see the raw count data, because then these names of these ERCC spike-ins will come out, right. And after normalization with spike-in controls, this is what we get. Okay. And you can see this is actually very different from what we have got earlier, right, across all different types of normalization, right. We still see, like for example, this sample or this sample, they look bit like outliers, right, they do not show very similar distribution across this data set, right. So, that is actually maybe the case, right, because there is some sort of variation that happened during the experiment that actually led to this kind of situation. Okay. So, the biggest question is now, which normalization method should you choose? We have discussed a lot of these methods, right, and the question is, which one would you choose, right. Again, there is no unique answer here, right, it depends on your application, what you want to do, right. If you want to do this within sample comparison, right, of course, then you have to choose a method that does this within sample normalization. But if you want to do this between sample comparison only, right, then you have to choose a proper method that will be able to handle this RNA composition bias across samples and also look at differences in other factors, right. So, it can model all these technical variation. So, you would have to choose something like that. But if you want to do both, right, then you have to probably combine some of these methods one after another, right. So, what I will do, I will not go into too much details of this, but this has been kind of searched around, right, and so here are some papers, right, some references, right, that you can explore to understand these issues better, right. So, these are some papers that actually compare different normalization methods for different applications, different sets, different types of analysis, and they found out different things. Okay. So, you can go into these references and you can explore more. Okay. So, one final thing that I wanted to discuss is something called batch correction. Okay. So, what happens in this when you want to combine different data sets of the same experiments. Okay. So, when you want to combine these different data sets, right, they can show different read counts, expression levels of same genes. Okay. And this is something that we very often see. Okay. And because these experiments are done on different time points, maybe years apart or in different labs, you tend to get different results because the experimental conditions are slightly different, etc. The people who are doing the experiment, they are also different, right. It also varies a lot based on the person who is doing the experiment. Okay. And again, as I mentioned, this is mostly experimental variation, right. Also the some of the conditions may have changed and which leads to these difference in expression levels. So, you can actually correct this using these tools, right. These are two tools I will mention, right. I will not go into all the details, but the first one is part of the lemma package in R. So, it feeds linear models to the data for analyzing the effect of batches, right. If you have batch data, you can actually analyze this using this package lemma. This was actually designed for microarrays, but can be extended to RNA-seq. And the second one is function called combat, again correcting for batch effects, right. It uses a Bayesian framework to adjust for batch effect. Okay, I will not go into details, but if you are interested, of course, you can explore this more. Here are the references for this class. To summarize, we have talked about very sophisticated between sample normalization methods, which can address this RNA composition bias and of course, other issues. And we have talked about two methods that can do this very well. One is the median of ratios method and the is TMM normalization. We have also talked about the normalization method using RNA spike-in controls, and they enable estimation of technical variation. We have seen the normalization data right after the normalization, and we have also talked about batch correction that allows us to combine data sets and compare across data sets. Thank you.